Okay, hello, welcome back again. So today we are going to cover chapter 27, and chapter 27 is all about bacteria and archaea. And you may remember from General Biology 1 that the all of the members of the domain bacteria and the domain archaea are actually prokaryotes. And we'll talk a little bit more. We'll review a little bit more in just a minute about what a prokaryote is. But we're talking about all of the individuals from domain bacteria and archaea. And remember that a domain is a higher level than a kingdom. And remember, there's three domains. In biology. One domain is bacteria, the other is archaea, and then the third domain is eukaryota, everything that has eukaryotic cells. So anyway, so as kind of an introduction here to get look at something interesting and to get you interested in the subject of this chapter, they mention something interesting about an area in Spain, a seaside area in Spain called Laguna Salada de Torrevieja. Laguna Salada in Spanish, I think, actually means salty lagoon, and Torrevieja is probably the name of the town there, and Torrevieja means old tower, I think. But anyway, so this area of Laguna Salada de Torrevieja in Spain is a lot of times it gets, for some reason, the seawater there gets saltier than usual seawater, and at certain times of the year the water turns pink, and it basically the water turns pink because of trillions of prokaryotes living there, and here you can see it. And this kind of reminds me of the one of the hot springs in Yellowstone that turns orange and green color because of the prokaryotes living there. And here's a good example of archaea. So this is the type of archaea then called a halobacterium that's here in Laguna Salada de Torrevieja here in Spain, that turns this salt water very pink during certain times of the year. So let's dive into prokaryotes then. A little bit of review from General Biology 1. They can basically live pretty much everywhere because different ones are adapted to all sorts of different types of habitats. They can live everywhere. They can live in places that are too acidic or salty or cold or hot for most other organisms. So since they've got such a great ability to adapt to a lot of different types of habitats, they are, well, also because they're kind of small, they are the most abundant organisms on Earth. In other words, they're the largest numbers of them on Earth. And that kind of makes sense since they're so small. Millions of them can be fit in very small places. You could probably have hundreds of thousands of them on the head of a pin, I suppose. So other than that, then what else about it is interesting? Well, so like I mentioned at first, the prokaryotes then that have prokaryotic cells, they're basically from two different domains, the bacteria and the archaea. And so the people that paleontologists that come up with ideas and things on evolution are thinking that possibly the prokaryotes were the first organisms to inhabit Earth, which kind of makes sense since they have very basic cells, very simple, very basic cells. Most of them are unicellular and exist as just single cells together, but some of them will actually join together and form colonies, and we'll see that a little bit later on in the chapter. And most of these prokaryotic cells are very, very extremely small, from 0.5 to 5 micrometers small. And remember, a micrometer is actually 1,000th of a millimeter. So it's much smaller than the usual range of 10 to 100 micrometers for a lot of eukaryotic cells. And remember, the eukaryotes, those that have eukaryotic cells, remember, are protists, fungi, plants, and animals. So the prokaryotic cells then have a variety of different shapes and sizes. And the three most common shapes are spheres, which they call cocci, rod-shaped, which they call bacilli, and then spirals. And so here you can see, some spherical cocci, some rod-shaped bacilli, and then some spiral. And a lot of these are called spirochetes, actually, because of their kind of crazy spiral shape. For example, Lyme disease is caused by a type of spirochete with the spiral shape. So now remember that a prokaryotic cell has a cell wall as the outer layer of it. And so the cell wall in these prokaryotes maintains cell shape, protects the cell from a lot of different things, prevents it from bursting if it's in a hypotonic environment. So if it's in a hypotonic solution, meaning the solution has some solutes that are less concentrated than the fluids inside the cell, it would actually burst. No. A hypotonic solution is a solution that has lower concentration of solutes than inside the cell. Sorry, just trying to remember there. And since it has lower concentration of solutes than inside the cell, water would tend to try to rush into the cell and cause the cell to burst, but a cell wall helps it to prevent it from bursting. Eukaryote cell walls, remember, plants and fungi can have cell walls as well, but these eukaryote cell walls are usually made of cellulose or chitin, but most bacterial cell walls are made of something called peptidoglycan. So peptidoglycan, so glycan means that the sugar polymer, the carbohydrate polymer part of it, cross-linked by polypeptides. So the peptido part refers to the polypeptides that are part of the molecule. And polypeptides are chains of amino acids, and if they're long enough, they could actually be proteins then. So the archaea, on the other hand, so the bacteria have cell walls that are peptidoglycan, but the archaea, on the other hand, have their cell walls have polysaccharides, sugars, in other words, and proteins, but they lack peptidoglycan, though. So that's one of the main differences between archaea and bacteria. And scientists use a technique called Gram stain to classify bacteria according to cell wall composition, because gram-positive bacteria have simpler cell walls with a large amount of peptidoglycan, but gram-negative bacteria have less peptidoglycan, and they have an outer membrane that has lipopolysaccharides. In other words, some lipids, a lipid part joined to some polysaccharide chains. So the gram-negative bacteria are bacteria that don't really soak up the gram stain very much, because the gram stain stains peptidoglycan. And here you show, here's a pretty good diagram then. So here's a gram-positive bacteria, here's a gram-negative bacteria, and you can see the cell wall of a gram-positive bacteria has a lot of peptidoglycan as an outside the plasma membrane, whereas the cell wall of a gram-negative bacteria has an outer membrane of lipopolysaccharide, and then a small, a thin peptidoglycan layer, and then the plasma membrane then. That's an inner membrane. And so the peptidoglycan then on the gram-positive bacteria traps the crystal violet in the gram stain, and that masks the red safranin dye. And so gram-positive bacteria look kind of purple, basically. So these cocci, these spherical cocci, are the gram-positive ones, and these bacilli on this slide, the rod-shaped bacilli, are the gram-negative ones. And in the gram-negative bacteria, then the crystal violet is easily rinsed away, and it reveals the red safranin dye that has dyed these gram-negative bacteria kind of a pinkish red color. So that's gram-positive and gram-negative then. And here you see close-up of the gram-positive bacteria and close-up of the gram-negative bacteria. Really nice drawings here and diagrams. And a good close-up view of the slide showing that. So a lot of antibiotics then can actually target peptidoglycan and damage bacterial cell walls then. And gram-positive bacteria are particularly susceptible to the type of antibiotics since it's got a lot of peptidoglycan in its cell walls. A sticky outer layer of polysaccharide or protein called a capsule is present in some prokaryotes. And the capsule allows it adherence or sticking to the substrate. Substrate basically means some sort of solid surface on which they can attach themselves. A solid surface of like the skin of an organism, or the bark of a tree, or a rock or something. So it allows it to adhere to a substrate, or maybe adhere to other individual bacteria. It can shield pathogenic bacteria from the host immune system. And pathogenic means causing disease. So patho is disease, and genic is to generate, to cause. So bacteria that cause disease then are called pathogenic. Or something that's a pathogen, like a virus, bacteria, protist, or even some sort of invasive worm, then that's a pathogen, something that causes disease. And here you can see, bacterial cell wall at the outer outside of the bacteria, and then the bacterial capsule that's the outermost layer. And then you can see this capsule on this particular bacterium is pretty dense, pretty thick, and you can see a lot of the lipopolysaccharides on the outer surface, kind of chains like on the outer surface, makes it look kind of a little fuzzy or something on the outer surface then. And here's a tonsil cell in someone's throat that it's attached to. So a great photograph taken under a microscope, and a lot of really great colors to that too. So this photograph is basically a work of art if you think about it. So a lot of prokaryotes then form metabolically inactive stages called endospores, which is kind of like a seed in a way, but it's dormant. When they say it's metabolically inactive, it means it's dormant basically. So it's sitting there dormant as an endospore. And these endospores, like a lot of seeds, can be viable and alive even in a lot of harsh conditions. Even if they're stored in a lot of harsh conditions, and sometimes even if they're stored for centuries, they can still remain viable. And here's a bacteria, and inside the bacteria there's an endospore that's that's surrounded by a coat there. Really nice photograph taken under electron microscope again. So some prokaryotes have kind of long, thin, hair-like appendages called fimbriae, and they allow them to stick to a substance, a substrate, or stick to other individuals in a colony. And then some of them also have long, thin appendages called pili, or sex pili. And these are longer than fimbriae, and they allow the prokaryotes to actually exchange DNA with other prokaryotes. And here you can see, a lot of fimbriae around the outer edge of a prokaryote. Again, really nice colors on this microscopic photograph here. So motility, the ability to move. So about half of all prokaryotes exhibit a behavior called taxis. So taxis is the ability to move either toward or away from some sort of stimulus, something that stimulated them. For example, chemotaxis is moving toward or away from a chemical stimulus. So how do they move? A lot of them have flagella. The most common structures used by prokaryotes for movement. And a lot of protists have flagella as well. We'll see in the next chapter that there are a lot of protists with flagella also. And flagella is plural, and flagellum is singular. And so they might be scattered here and there on the surface of the prokaryote, or they could be concentrated at one end or both ends of the cell. And the flagella of prokaryotes and eukaryotes are different. They're different in structure, and they're different in the mechanism of propulsion as well. And they're different in the molecular composition also. And so here's a diagram of a flagellum. So remember, flagella is plural and flagellum is singular. So here's a diagram of one flagellum on the surface of a prokaryote, and you can see that there's a long filament, and then there's a hook at the base, and a motor that makes this flagellum actually spin around. And here you can see, again, a microscopic photograph. So we're not just making this up. This is actually what it looks like in real life. Close up of the photograph then, and here's a video of the prokaryotic flagella that you can look at on your own time. So bacterial flagella then are composed of actually 42 different kinds of proteins that form the motor part, the hook part at the base, and then the long thin filament that's out there in whatever fluid that's surrounding this bacteria. Most of the proteins in the flagella are modified versions of bacterial proteins that have different functions elsewhere in the body. So these proteins that make up the flagella are not necessarily unique to. They're not only found just in the flagella; they could be found in other parts of the bacterial cell as well. So now prokaryotes, prokaryotic cells, usually don't have complex compartmentalization like eukaryotic cells have. And when they say complex compartmentalization, what they actually mean are the organelles, like the mitochondria and the Golgi body and lysosomes and things like that. Some prokaryotes do have specialized membranes that perform different metabolic functions, but they're usually infoldings of the plasma membrane. And so here's so the outer plasma membrane kind of folds in and forms different membranes. And so here there's actually respiratory membranes. Believe it or not, respiratory membranes inside an actual prokaryote. And then here's a photosynthetic prokaryote, and it's got thylakoid membranes. And now, if you remember from General Biology 1, you know that in plant cells, plant cells have chloroplasts, and the chloroplasts are what perform photosynthesis. And inside the chloroplasts, there are thylakoid membranes as well that have the photosynthetic pigments, like the chlorophylls, in them. And so there's also, basically here you see thylakoid membranes. It looks almost like a topographical map, actually. Kind of an interesting, really, really interesting structures here. These thylakoid membranes inside this photosynthetic prokaryote. So there are a lot of prokaryotes that are actually photosynthetic, and actually most oxygen produced on Earth is actually produced by photosynthetic prokaryotes that are in the ocean, believe it or not. So the prokaryote genome then has less DNA than the eukaryotic genome on average. Most of the genome in these prokaryotes consists of just one circular chromosome. So it's a loop, in other words. So whereas eukaryotic chromosomes are a long, one long linear molecule, kind of like a string of spaghetti, I guess. Kind of the genome of a prokaryote is a circular chromosome, a loop, and you'll see that in diagrams and pictures and things later on. This chromosome is not contained in a nucleus, though, in the prokaryotes, but it is located in an area called the nucleoid region of the prokaryotic cell, without a surrounding membrane. So typical prokaryotes also have smaller rings of independently replicating DNA called plasmids. So they've got one circular chromosome, but also they've got smaller rings of DNA as well that's called plasmids, that's outside of and completely separate from the chromosome of the cell. And here you can see chromosomes, parts of the chromosome then, and then plasmids. Here's a plasmid here, a small ring of plasmid. Here's another small ring here that's a plasmid also. So there are some differences between prokaryotes and eukaryotes in the DNA replication and in the transcription process and in the translation process. However, the overall process is pretty much the same. But these differences, though, allow people to use certain antibiotics to inhibit bacterial growth without harming the human cells. So prokaryotes reproduce fairly quickly, and each prokaryote cell divides by what's called binary fission. And generally, they can divide every 1 to 3 hours if they're under optimal conditions. The right conditions of temperature, the right conditions of concentration of the fluid around them, enough nutrients and things. But so typically they can divide every 1 to 3 hours. However, an E. coli bacteria, Escherichia coli, can actually divide once every 20 minutes, actually. So there's some range, some differences. So the key features then of prokaryote biology, they're very small, like we've mentioned. They reproduce by binary fission, like we've mentioned, and also they have short generation times. And so basically then, the prokaryotes do actually have considerable genetic variation. And there are 3 things that contribute to this high degree of genetic variation in the prokaryotes. Their rapid reproduction, the mutations that they have, frequent mutations, and also their ability for genetic recombination. And we'll go through each of these one right after the other in turn. So prokaryotes reproduce asexually, so their offspring cells are basically genetically identical then. And so binary fission basically is kind of the prokaryote type of mitosis then. So for example, Escherichia coli cells reproduce using binary fission, and they produce, so in each Escherichia coli, as it divides into two cells by binary fission, produces basically a copy of itself. Mutation rates are low, but mutations accumulate fairly rapidly because the generation times are short, and the populations are very large. So prokaryotes, as we've mentioned, have simpler cells than eukaryotes, but their rapid adaptation to environmental change indicates that they are actually fairly highly evolved. So if you do an experiment, if you do daily serial transfer, and serial transfer basically means transferring from one container to another, like these test tubes. So if you're growing prokaryotes in a test tube, and you take just 0.1 milliliters of fluid from the old test tube and put it into a new test tube with growth medium, and if you keep transferring on a daily basis, if you keep making up a new tube with new fluid in it, and a new test tube with new fluid in it, and a new transfer of bacteria, and if you measure the population growth rate relative to the original population, ancestral population, you can see that after a few thousand generations, the growth rate actually increases to quite a bit faster growth rate than what the ancestral population had. And so this shows that they can actually adapt and improve and change fairly substantially, over a few thousand generations. So then genetic recombination, this is another thing they can do. So the combining of DNA from two sources is genetic recombination, and that contributes to the genetic diversity of prokaryotes. So prokaryotic DNA from different individuals can be brought together by either transformation, transduction, or conjugation. And movement of genes among individuals from different species is called horizontal gene transfer. So a prokaryotic cell can take up and incorporate foreign DNA from the surrounding environment in a process called transformation. So they find foreign DNA floating around by itself and just take it up and transform themselves. Transduction then is the movement of genes between bacteria, moving them from one bacterium to another via phages, bacteriophages that we looked at in chapter 19. Remember, bacteriophages are viruses that infect bacteria. So here's a phage infects a bacterial donor cell that carries A+ and B+ genes. The phage DNA is replicated and phage proteins are synthesized, and then a fragment of bacterial DNA with the A+ allele is packaged within a phage capsule. And so now that phage capsule, that phage that has the A+ gene from the bacteria in its genome. And then the phage with the A+ allele affects a bacterial recipient cell, with the A plus gene. And this bacterial recipient cell happens to be A minus and B minus, let's say. And so the A plus gene crosses over with the A minus gene. And if it's able to, it can actually replace the A minus gene with an A plus gene. And so now basically you've just changed the genetics of this recipient cell then. And then conjugation, there's another way of producing genetic recombination among prokaryotes, which is called conjugation, a process where genetic material is transferred between prokaryotic cells from one prokaryotic cell to another. So in bacteria, then the DNA transfer is always one way from one cell to another. The donor cell attaches to the recipient cell by a pilus. Remember, pili is plural and pilus is singular. So it attaches with a pilus and it pulls it closer and transfers DNA through a structure called a mating bridge that was basically formed by that pilus. A piece of DNA called F factor is required for the production of pili. So here's a pilus then going from one bacteria to another. And so the F factor then could actually be on a plasmid. Cells containing an F plasmid function as DNA donors during conjugation. And cells without the F factor function as DNA recipients during conjugation. But the F factor itself is transferable during conjugation. So one that a bacteria that doesn't have an F factor could actually be given one. So provided some of the F plasmid's DNA is transferred, the recipient cell becomes a recombinant cell. And here you can see the conjugation and transfer of an F plasmid, and also conjugation and transfer of part of an HFR bacterial chromosome, causing recombination. And so we should actually look at this in more detail, though, in part 2 of the lecture over chapter 27. So we're going to end part 1 of chapter 27 right here at this point, but within the next day or two, I will actually record then part 2 of the lecture on chapter 27. So as always, every day check your email and also your Canvas for announcements so that you keep up with everything that's going on in the class. So thank you and have a good day.